Hello, everybody. Hi, Catherine. Thank you very much for this very, very kind introduction. So, as Catherine said, I will be talking today about big brain anatomy. And it's important for me to stress the fact that this course is really geared to beginners. So I've seen that some of the participants are colleagues of mine from whom I know that they know a lot about anatomy. So this really isn't a course for you, okay? This course is geared towards people with absolutely no knowledge on brain anatomy, but they're working with the big brain data set, or also to people with a small knowledge in anatomy. And what I would like to do in today's course is to familiarize you with the landmarks that are used by anatomists to navigate through the brain, and also to learn the terms that we use to describe different parts of the brain. And because in brain mapping, it's not just about localizing a certain structure, but about finding the correlation between structure and function, I will also be mentioning at different times functional aspects that we can associate with these brain regions. Now, of course, we all would have preferred to have a live meeting where we could all see each other, and well, you are lucky, you at least can see me. I've got a whole lot of little black squares with names on them. So I would like you to use the hands up function of in Zoom anytime. If you have a question and you want to interrupt me, PJ will be co-hosting with me, so she can also stop. She'll be monitoring the the questions and the chat room. And if you need to interrupt me at any stage, please don't hesitate to do so. I'd like to start off with an introduction to the terms that anatomists use to describe or define relative locations of brain structures in the brain. You will see that we often use several terms to describe the same structure. This doesn't make life easier, of course, for beginners. If you've learned one word and we use the other one, it can be at times confusing. So what you see in this slide here is a schematic drawing with a head with a nose and mouth, so that you know where the face is, and the brain located within this structure. And you can see that the words dorsal and frontal and basal or caudal are the ones that are used to define our relative position in relation to the brain, whereas superior, inferior, anterior, posterior would be terms that we use to describe relative position in relation to the entire body. And to make life just a bit more complicated, you must know that in the human brain we can define two axes. Okay, one of them is called the longitudinal axis of the forebrain, and that's the one that I'm indicating here with this red line. So the frontal, dorsal, caudal, and basal designations would be the one that would correspond to this axis here. And then we have a second axis, the one indicated by the blue arrow, and that is the longitudinal axis of the brain stem and spinal cord. And here, rostral, dorsal, ventral, and caudal have a different orientation. So, for example, the word rostral can mean two completely different directions depending on whether you are in the forebrain or in the brainstem and the spinal cord. Now, BigBrain is a 3D reconstructed volume of histological sections, and sections automatically mean that we have cut the brain. And when you cut the brain, you can do so in different directions or planes of sectioning. And there are 3 planes of sectioning that are used in a standard manner in brain anatomy. One is the coronal or the frontal plane of sectioning. It is the one that divides the brain into anterior and posterior parts. Then we have the sagittal plane of sectioning, which divides the brain into right and left parts. If the section that you're looking at is located in the middle of the brain and it divides it into 2 equal halves, then we talk about a mid-sagittal or median location of the section. And if this sagittal plane of sectioning divides the brain into 2 unequal parts, so if it's located more to the left or more to the right side of the brain, then we talk about a parasagittal location, okay? And the 3rd plane of sectioning is the horizontal one. You see here at the bottom left of the image. It's also called an axial plane of sectioning, and it's the plane that divides the brain into dorsal and ventral positions, or top and bottom positions. And on the right of the image, you can see this surface reconstruction of the big brain with the cutout that indicates these 3 planes of sectioning. Now, if you've already been working with BigBrain, you'll probably be familiar with this data set, but today I'd like you to meet a slightly younger version of BigBrain. This is a photographs of BigBrain that were taken before it was processed to create the 3D volume that you always work with. So you can see that BigBrain, it was obtained from a 65-year-old male donor. It weighs 1,392 grams, which is actually just slightly above the average mean weight of a male brain. That is around 1,200 grams. And the big brain is approximately 16 centimeters long when moving from anterior to posterior. And what I'm going to do now is zoom into this view here, which shows the left hemisphere, the lateral surface of the left hemisphere, and this view here, which shows the ventral or the basal surface of the hemisphere, just to highlight a couple of landmarks. Now that I've zoomed in and you've got a clearer view of these 2 images, you can see it becomes clear that the surface of the brain is not smooth, it's convoluted, and some parts of it are very heavily folded, such as the cerebellum, that is the part of the brain that I'm now highlighting in blue. Other parts are less convoluted. That is the part of the brain that we call the forebrain. And we have a part of the brain, it's more clearly visible on the basal view, that is practically smooth. And that's the brain stem, all of this part here. And then to the right of the brainstem, we'd have the beginning of the spinal cord. The brainstem enables the transfer of information between the brain and the rest of our body, okay? And so it's also involved in the control of automatic functions such as heartbeat and breathing. I'm not going to say any more about the function of the brainstem today. The cerebellum is involved in balance control and also in the control of voluntary motor skills. We also know that it plays a role in functional or in cognitive functions. However, this aspect of cerebellar involvement in brain function isn't to date well studied, probably because of the fact that because of its location, it's prone to a lot of measuring artifacts in functional imaging studies. And then I'll tell you more about the involvement of the forebrain in different skills later on at different steps during the talk today. Now, since we know that the cerebellum is located at the posterior part, the back part of the brain, and we also know that it is located underneath the forebrain, that helps us identify the rostral and caudal parts of the brain, and it also helps us identify the hemisphere that we are looking at, okay? And with this information about the relative location of the forebrain and the cerebellum, that is why I can tell you with 100% certainty that this here is the left hemisphere. And when we look at the basal view of the brain, we can see both hemispheres. And I can say that this here is the left hemisphere and that this here is the right hemisphere of big brain. This is something when that might sound trivial, but when you're working in 2 dimensions and you've got photographs, it's sometimes difficult to in your mind reorient yourself and rotate images. Okay, so with this, I'd like to start my first poll. I'd like you to participate and tell me, looking at this image of big brain, do you think that the asterisk that you can see here on the far right of the image, is it located in front of the left hemisphere of big brain or behind the left hemisphere of big brain, or is it in front of or behind the right hemisphere? Okay, and loads are clicking. Right, 66% of people have voted. Now, I do hope I'll get a bit more voting than when people go to general elections.
You won't.
Alan? Okay, 71% voted. We're almost there. Most of you, I won't tell you the answer because we've got a couple left. Hmm. 87. Right, okay. Oy, we've gone up to 80% now. It was stopped at, okay, well, anyway. So now the number of people voting has become quite stable. Fantastic, PJ has stopped this for me. Thank you very much. So the result of it was that most of you have said that the asterisk is located in front of the right hemisphere. So answer C would be the correct one. And that is indeed the correct one. It's not behind, because I had said that the cerebellum is located at the back of the brain. And you can see here that it's the back of the brain. Okay. We'll come back to another question a bit later on in the talk. Okay, I'm going to now, on the right side of the image, I've changed from the view of the basal surface of big brain to the dorsal surface of big brain, because here you can see a structure that wasn't visible earlier on. You can see the so-called longitudinal fissure. Okay, it's a deep cleft that separates the right and the left hemispheres. And I would now like to discuss the differences in these foldings between the forebrain and the cerebellum. Okay, so the surface of the forebrain and of the cerebellum, I mentioned earlier on, they're folded. And in the forebrain, we say that the bumps are called a gyrus, or if you've got several, it's gyri. And the gyri are separated by so-called sulci, right? So the sulci are the grooves in the forebrain, and the gyri are the bumps in the forebrain. The folds in the cerebellum, the individual folds in the cerebellum don't receive a name. So that's a good bit of news for you. That's one term less that you would have to learn. If we have a very, very deep sulcus in the forebrain, it's often called a fissure. And that is the case, this brain structure here. This is the lateral fissure. It's called the lateral fissure because it's on the side, on the lateral side of the brain, okay? And the lateral fissure isn't the only sulcus with a given name. There are actually most sulci and also most gyri in the brain have given names. And here I'm just showing some examples on the surface of the column 27, the single subject template brain of MNI space. But please don't worry, I'm not going to introduce all of these sulci to you in the big brain, and you're definitely not expected to learn all of these names in the big brain. I would just like to highlight a couple of sulci because they will help me make a point later on in my talk. Okay, now let's start again with a left-right question. If I tell you that this is a dorsal view of the big brain and that rostral is to the left side of the image and caudal or occipital is to the right side of the image, would you give me a thumbs up if you think that this is the left hemisphere? If now I've got my laser pointer on the left hemisphere. So do we have, yes, loads of hands going up. But I just saw that Ariane put her hand up, and I know that's, well, not cheating. Ariane isn't a beginner. Anyway, thank you very much for participating in this. So, right, we've got the left hemisphere here and the right hemisphere here. And what I've now done with these colored lines is I've highlighted the position of the central sulcus in red and of the precentral sulcus in right. Precentral because it is the sulcus that is in front of the central sulcus. Central sulcus, it's a very, we used a lot, or the anatomist that named it, so it used a very, a lot of imagination. It's called the central sulcus because it's the one that's located approximately in the center of the hemisphere, okay? Now, here you can see one aspect that Catherine mentioned in her introduction. Catherine talked about variability. And here you can see that variability doesn't just pertain to when you're comparing, so differences between different brains. We actually already find variability in certain patterns within one brain, but when we're comparing the left and right hemispheres. So here you can see that, for example, in the right hemisphere, the precentral sulcus has a branching close to the longitudinal fissure, but it doesn't have this branching on the left hemisphere. You can see that on the right hemisphere, the precentral sulcus is continuous in this, or at least at this dorsal location, whereas on the left hemisphere, we've got an interruption. And also when you look at the relative position of the precentral sulcus and the central sulcus, you'll see that the precentral sulcus merges with the central sulcus very close to the midline in the left hemisphere, but not in the right hemisphere. Okay, so these are things that tell us something about variability, and these are challenges that anatomists have to contend with when analyzing brain structure. The fact that the very many brains, they're all very similar. They all have a central sulcus and a precentral sulcus and a midline being this longitudinal fissure, and so on and so forth. But the patterns are slightly different, just in the same way as when we're looking at a person's face. Everybody has two eyes and a nose and a mouth and so on, but some people have green eyes or blue eyes, or their eyes are closer together, or they have a bigger nose, and things like that. With brain anatomy, we have the same problem. And just imagine how variable this folding pattern of gyri and sulci is, that not even monozygotic twins have exactly the same pattern. So the pattern of folding the brain is as individual as your fingerprint is, okay? So with this, I'd like to come to the next poll. Today it's the last one that I'll be setting today, okay? I'd like you to tell me, which one of these brains do you think is big brain? Would the correct answer be A, B, or C? So we've got a favorite winner. There's one brain that's way ahead of the other two. Okay, again, about 80% of participants have voted already. Yep, okay. So 63% of you think that answer A is the correct one. And then we've got 18% and 18% for brains B and C. Well, the correct answer is A. Brain A is the big brain. And if you compare them, I mean, this is just something I could spend hours looking at brains. It's just funny how, for example, big brain has this structure here in the left hemisphere that sort of like reminds me of a little of a ribbon, of a bow that you tie in your hair. Anyway, so what I would like to show you now is why is it important to be able to identify certain sulci? Why is it important for anatomists to do so? And now on the right side of the image, I've replaced the dorsal view of big brain with a surface reconstruction in which you can see the medial view of the hemisphere. So the one that is located in the longitudinal fissure. Okay, so this is what we call the medial view of the hemisphere, and this would be the lateral view of the hemisphere. And I've highlighted a couple of sulci in different colors. We've got the, I'm sorry, I'm having problems here with my mouse and the screens. You can see the central sulcus highlighted in red, the lateral fissure in black. On the medial surface, we've got the parietal occipital sulcus in blue, the cingulate sulcus in green. And then here we can also see a little red line because the central sulcus, it goes over the midline of the hemisphere and then encroaches onto the medial surface of the brain. And the central sulcus is a nice landmark because it indicates the border between the frontal and the parietal lobe. I've highlighted the frontal lobe in blue, the parietal lobe in green. And the other two lobes that we can see on the lateral surface of the brain are the occipital lobe and the temporal lobe. So they are highlighted like in purple and an orange color. And on the medial surface of the brain, we also have the limbic lobe, which is located around the corpus callosum. The corpus callosum is just, well, it's a collection of fibers, a grouping of fibers that connect the left and right hemispheres with each other and enable information transfer between both sides of the brain. So that both parts of the left and the right part of the brain can communicate with each other. The lateral fissure, it constitutes a good macroanatomic landmark to separate the temporal and the parietal and the frontal lobes. Here on the lateral surface of the brain, we have no macroanatomic landmark to highlight the border between the parietal and the occipital lobes. This is not the case for the medial surface. Here, it is the parietal occipital sulcus that serves as a very good landmark, okay? And the cingulate sulcus can help us most times identify at least the basic location of the border between the limbic and the frontal lobe. Now, this is one important take-home message that I'd like you to have for today, and that is the fact that not all sulci serve as landmarks or can be used as landmarks to identify structural or functional entities within the brain. but in some cases they can be of use. Now, why is it important to identify these different lobes in the brain? This is because each of these lobes can be associated with different functional domains, okay? The frontal lobe, for example, is associated with higher level cognition and with motor skills. So that's a nice term that people doing functional imaging do. You might wonder what is meant when we use such terms. Well, motor skills are, for example, the part of the brain that controls body movements. It's located at the posterior part of the frontal lobe, so close to the central sulcus. And the frontal lobe, when we talk about higher level cognition, it's the part of the brain that enables us to predict the consequences of our actions. It enables us to suppress, for example, socially inappropriate behavior. It's the part of the brain that would tell a European that it is not polite, for example, to burp at table, but it would tell an Oriental person that it's not a problem to burp because that would indicate that you enjoyed your meal. Okay. And the frontal lobe is also crucial when we want to produce so-called expressive language. So it's the motor control of language. It's the part of the brain that lets me produce sounds that you can interpret as language. The parietal lobe, on the other hand, is really important for the interpretation of touch. So the part of the brain where you can notice whether you've touched something, if you've applied a lot of pressure or just a bit of pressure, if you've felt something hot or cold, or you've been pricked by something, that part of the brain is the somatosensory cortex, and it's located within the parietal lobe. Also, it's located in the rostral part of the parietal lobe, close to the central sulcus. Whereas the posterior part of the parietal lobe, the part that borders the, so this part here of the parietal lobe, which borders the occipital lobe, it is very important in what we call visual-motor integration. It's the part of the brain that is crucial to control your movements when you are, for example, grasping a glass and bringing it to your mouth. So you have to know if you're applying enough pressure to your glass. If you press too much, you could break the glass. If you don't apply enough pressure, the glass would slip out of your hand whenever you move your hand to bring it to your mouth. You also want to bring your glass to your mouth and not, next to your ear. So it is really, an important part. For example, if you want to drink a cup of coffee or a cup of tea right now. This part of the brain is also important for you to be able to park your car smoothly, because it provides you with information of self-related spatial orientation, and it plays an important role in the knowledge of numbers and their relationships, okay? Now, the temporal lobe, it contains the so-called primary auditory cortex. So it is crucial for the perception and interpretation of sounds in general, and also in particular of language-related sounds, so language comprehension. When we move into the occipital lobe, we find that it's the part of the brain that is really involved in it contains the visual processing center. So it's what we see. It enables us to detect differences in light intensity, to identify colors, motion. It's divided into very many different regions with very specific functions. You've got the so-called face recognition area, which is only active when you see a face. It's not active when you are looking at a building. You've got other parts of the brain that are more object recognition areas, and they would light up in a functional imaging study if you presented the volunteer with an image of a building. And you've got other parts that are, for example, the word recognition areas, or the word recognition area, which is the one that enables you, for example, to look at this word here and recognize it as limbic, or even recognize it as a word that is part of your language. And then we've got the limbic lobe on the medial surface of the brain. Its more dorsal and rostral parts are extremely important for the processing of emotions. And its more ventral part contains a structure called the hippocampus, that I'll mention later on again, and which is the part of the brain which helps us with learning and memory processes, right? Now, I showed you the location of the central sulcus on this photograph of BigBrain. How could you find it if you had, or if you're looking for it in your volume? And that is, you could just do like a copy and paste, and then you'd have the central sulcus and BigBrain. But what I'd like to do with you now is for you to move over, whenever I tell you to do so, to the Atlas viewer using the link provided by Susanna in the registration site. And I want to give you a couple of tips that anatomists use to identify landmarks in the brain, particularly to identify the central sulcus when they don't have 3D data available, so when they only have sections. And I want to start with the horizontal plane of sectioning, because it's the one where this sign is it's the one with the most obvious sign that identifies the central sulcus. And radiologists have defined the so-called omega sign, so the Greek letter omega, because they say when you've got a horizontal plane of sectioning and you navigate from the top of the brain and you start moving down, right, then somewhere around the central part of the horizontal section, because it's the central sulcus that we're looking for, you will find a sulcus with a structure in it that looks like an inverted omega sign, okay? And that's what I've highlighted here. I'm going to remove the omega sign again, just so that you can see, right? So here you've got your gyrus, or your horizontal section without the location of the omega sign. And now here's the omega sign here. This is located on the hand knob, so the hand representation is located here. So that's how we know that this sulcus here, the one which contains an omega sign, we know that this is the central sulcus. If we move to the coronal plane of sectioning, you're going to have to use a bit more imagination, okay? Because here there's a sign that the radiologists call a hand with a ball. You've got to imagine that this gyrus here is a hand, and that this little gyrus here is a ball, right? As I said, you have to use your imagination. It can be more clearly seen in some brains than in other brains. And in the sagittal plane, the sign that radiologists use are the so-called two hands. So you look for two gyri that are sort of like interlocking. And that would be these two gyri here. So you've got to imagine your two interlocking hands here. The other tip that you can use whenever you're looking for it is that this gyrus that's at the front, this is the prefrontal gyrus. It is normally much broader, much more prominent than the sulcus, the gyrus that's behind the central sulcus, okay? So if you use these three tips, you'd be able to identify the central sulcus in each plane of sectioning in the viewer. Now, once you've done this in the 3 planes of sectioning, you can identify the site, which in the treasure map would be located with the X, okay, that this is the place at which your 3 arrows would intersect. And then from here, you can just draw your, or imagine that your sulcus moves down on the surface, and that's the way in which you would have identified the central sulcus on the surface of the brain. Now, there's one thing I must tell you, because I would now like you to switch to the browser to see if you can find these planes of sectioning in the left hemisphere, or which is what I've shown here, or in the right hemisphere. And there's one thing you must know. You're never going to be able to replicate this image that I'm showing you now, because it is due to the 3D structure of the brain. It's not possible to see these 3 views simultaneously in the brain, okay? I've just photoshopped them to be able to show them to you in this slide. So don't be frustrated if you can't see the hand with a ball and the 2 hands and the omega sign simultaneously. You'll have to navigate through the brain to be able to do this. So you'll have to open your browser and look for these structures. And maybe you can, if you have a particular question or really big, no, I just see here the tips aren't specific just to the left hemisphere. The right hemisphere also has an omega sign and a hand with a ball sign. But as I said, because of the 3D structure of the brain, you don't necessarily see these signs simultaneously on both hemispheres, and you definitely don't see them at the same time in the 3 planes of sectioning. And while you're looking at this, I can answer one of the questions that was posted in the question and answer section. Somebody asked if the limbic lobe is considered to be part of the neocortex, and why yes or why no. The answer to that question is yes and no. Parts of the limbic lobe are neocortex and parts are not neocortex. And I'll go into that in more detail later on in the talk. I've got a slide where you can see that. So has somebody already been able to find the omega sign in the right hemisphere?
So, Nicola,
Yeah.
Just a quick note. The first question, I believe, refers to your poll when you showed the 3 brains and you are asking which. So someone is asking, what are the other brains called?
They don't have, so BigBrain has a given name. It's called BigBrain. The other brains, they're just brains, like, you know, they're not called, they don't have other names. Is that the question? Like they would just be, we would identify, you know, different brains with an anonymized brain ID. Okay, so let me see. I hope I'm sharing screens with you. And well, I know I still am sharing screens with you, but I hope you can now see the Atlas viewer and not my PowerPoint presentation. PJ, can you? Oops.
We see the Atlas viewer.
Okay, you see the Atlas viewer. Now it's closed. Okay. Now, yeah. If I click here, somebody asked if these structures can be seen in the right hemisphere. I'm going to move up in the brain because I said that to find the omega sign, it's best to move to the top of the brain. So I'd be up here, my horizontal section. Then I'd start moving down till I found a structure that reminds me of an omega sign. And on the right hemisphere, here is where I would have my omega sign, right? If I continue moving down, then I see the image that I showed you earlier on. You can see the omega sign here, okay? And the right hemisphere, where have I got my 2 hands? Oops. It's not always easy. Oh, come on. So can you see here, the right image? We've got a very wide gyrus and a narrower gyrus, and with imagination, you could see 2 interlocking hands here. And this is also a position at which we could say that this is the hand and this is the ball, okay? So that would be how we can find our right. So then let's get back to the presentation. Okay. Because now that up to now, what I have done is I've talked about structures that are visible on the brain surface and take-home messages, things that I want you to remember. I want you to remember that that the forebrain, the cerebellum, and the brainstem are the major or are the major components of the brain. I want you to remember that the brain surface is that the folds of the brain surface are called gyri, and that they're separated by sulci. I want you to know that only at times some sulci or that only some sulci serve as highly reliable functional and microarchitectonic landmarks, and that although the hemispheres were originally divided into lobes just as structural landmarks, we can also relate these anatomical structures with different brain functions. Okay. And now I'd like to move from surface of the brain to structures that we can see in a brain section. Now, what you see here is a coronal section through the frontal lobe of big brain. So down here at the bottom left of the image, we've got this lateral view of the left hemisphere of big brain, and the lobes are still colored in according to the previous coding in the previous slides. And what I've done is I've highlighted the part of the section that belongs to the forebrain in blue, and the part of the section that constitutes what we call the pole of the temporal lobe, that's highlighted in yellow. And because it's a coronal section, then we know that the dorsal is to the top of the image and ventral to the bottom, and we've got the left and the right side of the section, okay? The left and the right side of the brain. And what you can see here is that the gray values in the image, they aren't equally distributed throughout the section. We've got approximately a 3 to 5 millimeter stripe on the surface of the hemisphere with darker gray values than this central part here. And we also have structures here in the center of the hemisphere, which are more darkly stained again than this part here, okay? And these darkly stained parts or more darkly stained parts of the brain are called gray matter, and the paler parts are called white matter. And just so that you know why we call these two parts of the brain gray matter and white matter, I'd like to introduce you to the neuron, which is the main cell type that we find in the brain, okay? And a neuron has different parts. It has the so-called cell body, okay? It's got dendrites and it's got an axon, which is can vary in length depending on the type of neuron that we're looking at. And it is covered, it's myelinated, it's covered in a so-called myelin sheath. That's what's indicated here by these thicker black lines. In this part on the axon than, for example, in the dendrites and in the cell body. And then at the end of the axon, we've got the so-called synaptic terminus. Okay, and this is important to know because this structure here enables the neuron to receive information. So the dendrites of one neuron receive information via synaptic terminus from a so-called presynaptic neuron. The information then travels through the dendrites to the cell body and then along the axon, where these synaptic terminals can then make contact with other neurons, okay? And that's the way information is passed on in the brain. And the thing is that the dendrites and the cell body and the synaptic terminals, they're located within the gray matter of the brain, whereas the axon is located in the white matter of the brain. And this difference in color is mainly due to the fact that myelin is a fatty substance and it is white, okay? Whereas in the gray matter, we have the cell bodies and we also have blood vessels. So we call it gray and white matter, not because the image that you saw, it looks gray and white. It actually already looks gray and white in the native brain before we even do any section or any sectioning or any kind of staining, okay? Right. So back to the coronal section, you will see that I've highlighted part of the gray matter with a red line. That is because the gray matter that is located on the surface of the brain is called cortex. It's from the Latin bark. It's like the bark of a tree. And the gray matter that is buried inside the forebrain is called a subcortical nucleus. Alan, you have a question. I think Pete, yes.
I'm sorry, I was just wondering the old story, how many neurons versus how many glia in the brain? This is very controversial. What's the current thinking about that? Used to say 50 to 1, and then they said 10 to 1, and now it's down to 1.
They said 10 to 1. That's the number that you find in all, or not, well, yeah, in most anatomy books. God, I don't have a slide on that here, but I can put one in afterwards. The fact is that the ratio between neurons and glia changes depending on what part of the brain you're in. So you've got parts of the brain where the ratio is much higher. For example, you've got a much higher ratio in the white matter, a much higher neuron to glia ratio in the gray matter than in the white matter, and it also varies depending on whether you're looking at the forebrain or the cerebellum. I do have a slide with that for another talk that I gave, and I can bring it up at the end of the talk. It's something that was, the 10 to 1 ratio is something that appeared for years in as I said, in anatomy books, and it was never contended. And it was recently, there was a very nice study by Susanna Herculano Hosel with counted these differences.
Thank you.
Right. Okay, so we've got cortex. It's gray matter located in the surface of the hemisphere. And we've got subcortical structures. Those are, it's gray matter located within the hemisphere. And what you must know is that depending on where we are on the brain, we can identify different subcortical nuclei. And on this section here, you can see the caudate nucleus, the putamen, and the nucleus accumbens. It's sometimes also just called accumbens. And then we've got the claustrum. And the caudate nucleus and the putamen, they are sometimes also joined into a structural entity called the striatum. And the striatum together with the nucleus accumbens build the so-called basal ganglia. Okay, so this here, this grouping of caudate nucleus and putamen into the striatum, that is a structural entity. It's called striatum because striatum comes from Latin and means stripe, and that is because during brain development, the putamen and caudate nucleus, or the putamen and caudate nucleus, they're actually together, and then the axons of neurons that are located in the cortex, mainly in the motor cortex, their axons want to find the shortest path down to the brain stem, and that is, it basically runs through this entity which is originally built by the caudate nucleus and the putamen when they're together. And then these axons, they push through it, and that's why they separate the caudate nucleus and the putamen, which then in the adult brain are still joined to each other via the so-called gray bridges. Okay. Now, here we have two rostral sections, again through the left hemisphere of the big brain. The one on the left is located, or they're called rostral sections. They can also be called coronal sections. Okay, the one on the left is located more rostral to the one on the right of the image, so that's why I've labeled them with rostral and caudal section. Okay. And what I'd like to do here is, again, a bit of interaction with you. And what I want you to do is to use zooms, emojis, because I'm going to place my laser pointer on a part of one of these sections, and I want you to then tell me if you think that that the laser pointer is on a cortical structure or if it's on a subcortical structure. So let's just start. Let me put my laser pointer here. Give me either a heart or a thumbs up. Yes, we've got a thumbs, no. Hand up. Is that a hand up because it's a question, or a hand up because it's questions and answers? How is that working with hands and hearts? Okay. At the bottom of your image, you should have emojis. You should have a little smiley. PJ, do the participants have a little smiley? No. PJ, could you give the participants a smiley? Is that possible? Right, yes, somebody has a heart. So here I'm back. Right, so yep, my laser pointer was on cortex. Yes, very good. Now if I move my laser pointer here, would you say that this is a heart or a thumbs up? Good. A hand. I'm going to take that as a hand, right? Okay, well, this structure is a subcortical structure, and it's actually called, it's a nucleus within the thalamus, okay? And if I put my mouse or the laser pointer here, would you also give me a hand, or would you give me a heart? Right, a hand. Okay, yes. This is also a subcortical structure. It's also part of the thalamus, okay? And the thalamus is composed of different nuclei. All of them together have one same global function, which is to, Olga, I saw you just putting your hand up. You're an expert. The thalamus, it's a so-called relay center in the brain. It passes information on from our sensory organs to the cortex, okay? And it's also a part of the brain that integrates information. And it plays an important role in the regulation of our consciousness, alertness, and also our different phases of sleep, okay? So here we've got the thalamus as a subcortical structure. Okay, and if I go here, is that a cortical structure or a subcortical structure? Go on, somebody give me a hand or a heart, please. I can't see what's happening. Yes, so hands, wonderful. I got a hand. No. PG, are you getting hearts?
Yes, there are some hearts.
Yeah, but they're not supposed to be hands. I mean, so I haven't explained myself. So if you put, oh dear. Now, if people put their hands up, does that mean that this is a subcortical structure? It's too complicated with the emojis. Right, well then we'll just do it in a different way. Hands up if you think that this is a subcortical structure. Good, fantastic. We've got hands, and it is, because it's the caudate nucleus, which you just saw earlier on. And hands up if you think that this is a subcortical structure. Good. We've got hands up. This is the putamen, which we saw again. And hands up if you think that this is cortex. Okie dokie, right. Now, do you think that that is cortex? Hands up if you think this is cortex. No. Okay. Now, let's go here. Hands up if you think this is a subcortical structure. Loads of hands. I like this. It is a subcortical structure. It's what we call the globus pallidus. Okay. Now, remember I told you just a second ago that the caudate nucleus and the putamen build a structural entity which we call striatum. Well, the putamen and the globus pallidus, they build a functional entity which we call lentiform nucleus. Okay, and the striatum and the lentiform nucleus, they play a very important role in motor control. Now, the amygdala, would that be a structural cortex or would that be cortex? Hands up. Some hands are going up. No, the amygdala is a subcortical structure. Okay, and it is also a collection of nuclei, just the same as the thalamus is, and it plays a really important role in the control of emotion. So for example, fear. And now the final structure that I want to look at right now. This here, would you call this a subcortical nucleus? Hands up if you think it's subcortical. Okay, some hands are going up. No, again, the hippocampus is a cortical structure. Okay, I mentioned it earlier on as a part of the limbic system that's involved in learning and memory. And the hippocampus it received the name hippocampus in 1564, and it was given this name by an Italian anatomist because he thought that the macroanatomical shape of the hippocampus, it reminded him of the fish of the genus of the seahorse. Now, if we have a coronal section and we create a cutout to zoom into the hippocampus, you can see that the hippocampus is made up of two gyri with interlocking C's. Okay, so we've got a small letter C and a capital letter C, and the small letter C is located on the dentate gyrus, and the capital C is part of the so-called corno ammonis region. And these colored lines, they highlight the different layers within the hippocampus. And that brings me to the last part of my talk today, which is the one of layers, cortical layers. You must know that within the cortex, cell bodies are arranged in horizontal layers, and that in the largest part of the brain, the cortex, that's what's indicated, or the cortex contains 6 layers. And that's the part of the brain that we call neocortex or isocortex, and which is highlighted in white in this slide here. And then we've got a part of the cortex which has either a very poor lamination, so basically no layers at all, or a very complex lamination with up to 12 cortical layers. And that is the part of the brain that it's the oldest part of the brain from the evolutionary point of view. And in the human brain, it only occupies about 3% of the surface of the brain, and it's what we call the allocortex. And then between allocortex, which is highlighted in red, and neocortex, which is in white, we've got a so-called transition zone, which is called mesocortex, and it's in blue here. Now, concerning the question earlier on as to whether the entire cingulate gyrus can be considered neocortex, yes or no, the answer to that is that the rostral portion of the cingulate cortex, where we find Brodmann area 24, that part of the cortex is so-called mesocortex. The part that we see on the surface, right? It does not have a layer 4, so that's why it's not neocortex. The posterior part of the cingulate cortex, where we've got Brodmann's area 23 here, we have our clear 6-layered lamination, so this posterior part is neocortex. And when we look at the part of the cingulate gyrus that is that directly abuts the corpus callosum, which is highlighted in gray here, and is the part that is buried in the callosal sulcus, that part in the rostral part of the cingulate gyrus is allocortex. but in the posterior part, it would be mesocortex, okay? So the transition between allocortex and neocortex. So I hope that answered the question that you posed earlier on. Now, criteria that we use for cytoarchitectonic mapping within the neocortex, so within the part of the brain that was highlighted in white in my previous slide, is basically the number of layers that can be identified, because I said we've got 6 major layers. But in some parts of the brain, some of these layers can be subdivided, and that's when we can see, for example, layers 3A, 3B, and 3C, sorry, in some parts of the brain. We've got 2 parts of the isocortex that are special in the sense that their layer 4 is very different from layer 4 in every other part of the brain. Those are the primary visual cortex and the primary motor cortex, okay? They constitute 2 extremes. And if we zoom in to this part of the section from BigBrain that is highlighted by these red frames, we can see that the distribution of cell bodies in the primary visual cortex differs considerably from that in the primary motor cortex. In the visual cortex, you can see more stripes, horizontal stripes, than in the motor cortex. And this is basically or mainly due to the fact that in the primary visual cortex, layer 4 is subdivided into 4 sublayers. This is the only part of the neocortex where layer 4 can be subdivided, okay? We also have a very prominent layer 6A. So it appears as a very dark stripe here, okay? These are cytoarchitectonic characteristics that enable us to identify this part of the cortex. And in the primary motor cortex, we've got the opposite situation. We've got the problem that layer 4 is not visible at all because cells from the adjacent layers, so layer 3C and layer 5A, they invade layer 4, and that's why we can't see the granule cells that compose layer 4 in the adult primary motor cortex. That is one characteristic that we use to identify the brains. The other one is the fact that layer 5B has these very, very large cells. They're the so-called giant Betz pyramidal cells. And this is also the only part of the brain where we can find them. Now, when we've got primary areas such as the primary visual cortex, it's quite easy to identify the border between primary areas and secondary areas. And that's what I want to show in this slide here. To the left, we've got the primary visual cortex. To the right, we've got the secondary visual cortex. On the left, you can see then these red arrows highlight the border. And you can see it because, for example, this dark stripe of layer 4C, it disappears at this position here. We also see this prominent layer 6A, it disappears at this position here. It becomes less prominent because to the left, we've got more cells. We've got a higher cell packing density than to the right. But in layer, or in area V2, sorry, in layer 3, we've got some large cells that don't appear in area V1. These are so-called Maynard cells. So these are factors that we look at to define this border here. The problem is whenever we move away from primary areas, it becomes very difficult to identify cortical borders based on on these differences in laminar distribution patterns. And that's why it was so important that Catherine Catherine Ammons, for those of you that don't know her personally, and Carl Sillis, they established and developed a quantitative and statistically testable approach with which we could identify cortical borders. Now, this is something that was developed in 2D, and now with BigBrain, and this is my second last slide. It's a 3D volume. It poses, it gives us a lot more chances of mapping, but it also brings a whole load of challenges. And I think, for example, that Konrad, who will be giving a talk tomorrow, has done a wonderful job with this first whole brain automatic detection of quantitative quantification of cortical layers in the 3D volume that constitutes the big brain. So this is one of the examples of what can be done with the 3D brain volume.